Welcome to Fermilab's 2021 Virtual Earth Day celebration. I'm your moderator, Sue Steyer. We'll begin Mark Donnelly's presentation, Dragonflies and Damselflies of Fermilab. But first, we ask that you please mute your audio and turn off your camera. Mark will answer questions at the end of the presentation. We invite and encourage you to type your questions in the chat box found on the bottom banner of your screen. This presentation is being recorded. And now, here's Mark.
Thank you, Sue. It's a pleasure and an honor to be asked to speak to you today at Fermilab's birthday celebration. Like Sue said, I'm Mark Donnelly. I'm a volunteer with Fermilab Natural Areas, and Fermilab Natural Areas, called FNA for short, is a nonprofit organization that takes care of the lab's ecosystems, primarily by harvesting and planting seeds and removing invasives. But they also create a structure for monitoring wildlife at the lab. And that's what I did for 5 years, monitoring the dragonflies and damselflies here. I first got interested in aquatic insects at a course at the University of Minnesota's field biology station at Lake Itasca, source of the Mississippi. It's also where I met my wife. But things change in life, and I drifted towards biochemistry and spent 30 years doing research on microbial enzymes and metabolism, mostly related to the environment. But after retiring, my interest in aquatic insects resurfaced, and specifically interest in the dragonflies because of the beauty and dynamic nature of the adults. So I'd like to begin by just sharing some of the beautiful creatures that I've seen here at Fermilab. This first, the ruby meadowhawk, you'll probably recognize as the poster boy for the presentation. It's a common species that lives in meadows near woodlands and reproduces in woodland ephemeral ponds. The rainbow bluet is named, you can see, because somewhere on its body, it has almost every spectrum of the rainbow. It's a rare species, not common in the Chicago area, and a real delight to find. Here are two species. On the left, the diminutive dragonfly, the eastern amberwing, perched as it often is on the stalk of a flowering aquatic plant. That's the actual flowering stalk of that plant. And on the right, a fairly rare damselfly, the eastern red damsel. This is a species that prefers seeps and soggy ground, and I'm going to mention it later in the talk. Here's one of the giants, the Cyranodarner. It's named for the literary anti-hero because of the large white schnoz that it has. It's flown up into a tree to eat an insect that it caught on the wing. This is the slender spreadwing. It's obvious where it gets its name. It's a male, and the males have this very long, slender abdomen. It's a species that lives in swamps. This was photographed at the very periphery of the lab, almost on Butterfield Road. Another large and quite rare species, this is the flag-tailed spiny leg. I saw this species next to a pond near the Letterman site, actually it was near the Wilson building. And it was the first time I'd seen one in the Chicago area. I've seen them regionally, but not in Chicago. So it was quite a find. I'll talk about it more later as well. I don't know what species this is. It's a female meadowhawk, and they can be very difficult to identify. To identify them, you have to net them and look at them with a magnifying glass, then let them go. But I decided to leave this one alone because it looked like she had a hard enough life already. Look at the tattered wings. They're not as impressive as the male adults, the bright red colors, but actually they have lots of subtle browns and yellows with hints of greens. And I often overlook them, I think, because I'm just too much attracted to the others. But if I take my time and look at these carefully, I really appreciate their beauty. This is a citron forktail, a tiny damselfly, barely an inch long, with colors of orange, lime, and lemon on it. It's another species that prefers soggy, seepy ground, and I'll mention it again later in the talk. I caught this shadow darner on the wing over one of the native natural ponds or streams, I mean, the Indian Creek in the lab, they're one of the mosaic darners, which are large darners that are named because of the blue and green and yellow spots and stripes they had on them that almost look like tiles inlaid into a brown background. And finally, the ebony jewelwing. This is a beautiful, huge damselfly. This is a female. The males have a iridescent green abdomen, but both have these dark jet black wings. And it's a very special species. It's one of the rare species that actually performs courtship, where the males court the females. But turning to Earth Day, this is a screenshot from Earth Day's website proclaiming their theme for this year, Restore Our Earth. And Fermilab and FNA are helping to restore our Earth. Fermilab is also, FNA is a 503(c) nonprofit organization that's able to receive donations. And with the money they get, they hire student interns, sometimes contractors, but they also buy trees that are planted on Earth Day. This is Penny Casper, the president of FNA, and her husband Peter, the lead bird monitor on site, planting a tree on a previous Earth Day. This year, unfortunately, there can't be any on-site celebration for you to come in and help. But I would recommend, if you can, do it yourself. Last year, I bought 2 native trees, an ironwood and a musclewood. They're short understory trees, and I've planted them and they're doing well. So you may want to do your own Earth Day celebration at home. FNA also educates. They have programs and they lead field trips. This is a shot from a field trip a couple years ago, a prairie field trip, where we were surprised by over 100 people arriving. Some of the staff, the FNA staff, had to act as surrogate guides to help Wally Laverniere, who was the main guide for this field trip, because not everybody could hear him. Hopefully you saw Wally's presentation of a field trip in the woods that are right to the right and behind this group earlier this morning, showing the spring ephemeral wildflowers. It was very enjoyable and very chilly for him, I imagine. I'm going to present a field trip of sorts later on, and I'll talk to you about that when we get to that point. But I wanted to talk a little bit more about Earth Day, its theme, and Fermilab, because over the last 50 years, Fermilab has both preserved and restored the Earth by protecting lands from development and creating new habitats. This is an aerial photograph of the site in 1939. It was purchased from the website historicaerials.com. It's a very interesting site. I recommend you go there and you can find your house before it was built, while it was being built, and afterwards if you're lucky. But what's striking here is the land is almost entirely farmland. You can see some remnant woodlands here. On the right, you can see the curve of the west branch of the DuPage River. And here, this line, that's the 2-lane rural highway, Route 59. But now, the transformation is drastic. You can see on both sides of the lab, east and west, subdivisions butting up against the lab, and on the north and south, industrial parks. So this is the aspect where Fermilab has preserved the Earth from development. Another thing that you may notice about this is if you go back, how the texture of the land has changed from this variegated farmland to a different type of texture, seen especially in these areas on the left and in the center of the ring. Here's a map of the areas defined by land management units, and you can see what's going on here. The dominant feature in the ring and on the left in orange are prairies, and these are prairies that have been reestablished over farmland. There are also woodlands, savannas, shrublands, marsh, but Wally talked a little bit about prairies earlier and mentioned Prairie 6. This is Prairie 6. It's located in the southwest quadrant of the ring, and it's actually spectacular. You can see all the flowers going off into the distance with the grasses beginning to grow up. This happened because of the efforts of 2 people, Robert Betts, a teacher at the local university who was obsessed with prairies and wanted to restore them, and Robert Wilson, the lab's first director, who said, go ahead and do it. The other feature I want to point out here, though, that's relevant to dragonflies and damselflies is the open water and the streams and ditches that you see at the bottom. All of these blue areas are now open water. And if you saw Eric Milan talk on Monday, you'll realize that these are part of the lab's cooling water system. The accelerators used to study particle physics generate a lot of heat, and water is vital to their dissipating that heat. It's also vital to dragonflies and damselflies because, whoops, I lose this. They're aquatic insects, primarily. We see the adults above water, but they have a life cycle that goes into the water living as larvae, which are called nymphs. And that's most of their life. They spend months to years underwater as larvae, then emerge and spend just a few weeks as adults, the ones we see and enjoy so much. This introduction of water here has created a huge amount of habitat for the dragonflies and damselflies, and they've exploited it. These are two examples of parts of the cooling water system. One on the left is a pond inside the main ring. You can see the aquatic plants, the newflower water lily in the front, the yellow water lily, and in the back, the American lotus with the white flowers flowering, and the building, Wilson Building, named and designed by Robert Wilson in the distance. And on the right, that's a ditch, just a ditch where two cooling water streams come together and flow out. It's quite wide, and you can see on the left and foreground emergent aquatic plants and submerged aquatic plants, all native. This area supports a surprisingly large number of dragonflies, damselflies, also butterflies, other species, leopard frogs, and once I even saw a pair of mink there, probably looking for the frogs. What I'd like to do now is divert and do a field trip. It's a virtual field trip, I call it, because whereas Wally's was virtual, he was there. You weren't, but he was. This one, I won't be there either. It's actually a compilation or a cobbling together of 4 educational videos that I've made over the years about dragonflies and damselflies. There may be a few rough moments between the sections, but I hope not. And it does amount to what you see in good field trips. In field trips, you go out looking for, you know, you're going to spot some things, but when you're looking at dragonflies and damselflies, you hope to find some exciting events, some educational and interesting occurrences. This video essentially comprises 5 years of primo field trip events into 16 minutes. So I hope you enjoy it. But to stream it, I have to change my sharing settings, so it'll take several seconds for me to get over to it and run the video. It won't run right away. So be patient, please.
Every summer, along with the birds and butterflies, dragonflies reappear to brighten our lives. Some fly in from afar. A few species are even known to migrate. But most arise nearby from local waters, and that's where they're most likely to be found, over ponds and lakes, along streams and rivers, at the edges of swamps and marshes. And when you're watching the dragonflies, you're sure to notice their smaller, slender cousins, the damselflies. Damselflies stay lower, flying slowly but precisely through the vegetation near the water's edge. Both groups have a large head with huge eyes, a body, the thorax, that carries the wings and legs, and a long 10-segmented abdomen. But the difference between the two groups is usually obvious. Damselflies are smaller, thinner, hammer-headed, and hold their wings back when perched. Even the spread-winged damselfly holds them back partially. The spread-wing also illustrates the core difference between the two. Damselflies' wings are the same shape. Compare that to this dragonfly's wings, which aren't the same, the hind wings being much broader. And note how dragonflies hold their wings out flat when perched, and are larger and stouter than damselflies, with a bulbous head. Dragonflies also roam far from water, especially when younger. You'll see them in old fields, in your gardens, in forest clearings, warming themselves amid the dew on cold mornings, or in prairies. See how the light projects through the wings of this eastern amberwing, like through tiny stained glass windows. Damselflies also range far from water. This damselfly is cleaning itself desperately, possibly removing residual spider's webbing left after a close call. This is a band-winged meadowhawk hiding behind that stick, a female. She's paused from her wanderings to feed away from the water before heading back to reproduce. Dragonflies grab their prey out of the air and start eating them while still on the wing, one pair of small mouthparts manipulating the prey while another chews off mouthfuls. See how the third, the outer pair, their lips, ratchet back and forth. Dragonflies may take larger prey as well. Here a female eastern pondhawk dines on a male meadowhawk. Males also range away from the water when they're young, like this blue dasher, eating, pooping, eating. The good life. But it must end, for soon he must return to the water. It can be tranquil, with just a few males patrolling the pond or staking claims along a woodland stream. But it can also be chaotic, with dozens of males of different species contesting the territory. Males patrol the water's edge, trying to claim part of it for themselves, fighting and feeding, but mainly waiting for females to show up, because this is where dragonflies mate and lay eggs. When a female arrives, she quickly links up with a male in a wheel-like formation where mating takes place. And right after mating, they start laying eggs. Here a female taps eggs onto the water while her mate flies nearby, guarding her. Here you can see what's going on. The male first grabs the female behind her head with a pair of claspers at the end of his abdomen. She then arches her abdomen forward so that a segment near the end, where eggs are produced, contacts a segment near the front of his, where he earlier placed a satchel of sperm. The process is exactly the same with damselflies. Damselflies also lay eggs immediately after mating, but look at the spine at the end of her abdomen, called an ovipositor. She's injecting eggs into the vegetation. All damselflies do this, as well as some dragonflies. Notice the sensory appendages at the end of this spreadwing's abdomen. These probe the surface of the grass and help her position the egg optimally. When she's finished, you can see the small bumps left by the injected eggs. Much more commonly, eggs are laid in floating or submerged vegetation, seen here with an eastern forktail in a spring pond. Among the dragonflies, only the darner and petaltail families have a true ovipositor. This is a pair of the common green darner, the species you'll see all season long, ovipositing eggs in vegetation at the edge of a small pond. The rest of the dragonflies lack a functional ovipositor. Instead, they release eggs through a pore or spout near the end of their abdomen. Here's a female white-tailed skimmer seen in slow motion, depositing eggs directly into wet moss. And here's her male, guarding her in what's called non-contact guarding. Some species remain attached while laying eggs, such as these calico pennants. And in a variation on catch and release, pairs of saddlebags sweep across the water until the male releases the female, then catches her after she's dropped down to put eggs in the water. Most casual of all, meadowhawks simply flick eggs out of their abdomen. Watch this. Dropping them onto vegetation at the edge of the body of water where they've bred. There goes her male. The reason males guard their mates is because their eggs aren't fertilized at mating, but only now, as they pass out during egg laying. Other males attack females laying eggs because they can still gain precedence over this clutch of eggs. And that's why their mates guard them, either by hovering nearby or staying attached. And what about courtship? From what you've seen, it would seem a female's only choice is to fly away. But some females are courted. Ebony jewel-wing females, distinguished from males by the white dot at the tip of their wings, coexist unassailed at the edges of streams in the presence of numerous males. Here one perches near the top center of the frame. She flies out to feed and back without being attacked. If a male does approach her, she warns him off by spreading her wings. Males claim and defend patches of vegetation suitable for egg laying and confront other males with ritualized flight displays. They advertise their sight to females by raising their abdomen to expose a white patch. If one shows interest, they cross and vibrate their wings, seen here in slow motion. And if one flies closer, they lift off in a vigorous flight display. The male continues to display along the water's edge as the female watches passively, not spreading her wings, until she flies up into the adjacent vegetation. He then follows her and continues the courtship dance until she allows him to land on her wings, walk down them, and clasp her. After a minute or two, they separate and the male leads her back to the egg-laying site. There he continues to display, both for her benefit and to ward off other males. And she begins to lay eggs. Other rare examples of courtship exist, but none as elaborate as the jewel wings. You may be wondering, with all this emphasis on water and eggs being laid in water, what happens next? The most important thing to realize about these insects is that they are, in fact, aquatic insects. They live for months or even years underwater, but only a few weeks above. Here you see eggs of a ruby meadowhawk, and look, to the right, one of them is hatched to produce the aquatic larva or nymph. This little creature is less than 1/8 of an inch long, but will grow over many months to emerge finally as the adult dragonfly we know so well. Even in small aquaria, nymphs can be difficult to see. To grow, they must break out of their external skeletons, swell, then harden again in a series of molts. Add a little mud, and they're almost invisible. But they clean up well and are quite beautiful in their own way, with fine sensory hairs on their legs and often spines on their sides to dissuade fish. They jet around by expelling water from a cavity in their rear end, which is also where they extract oxygen from the water to breathe. Damselfly nymphs are smaller and thinner, with gill-like blades at the end of their abdomen that aid in breathing. They wriggle like fish to swim. All nymphs are voracious predators, capturing their prey with a long, hinged lower lip, the labium, that flashes out, grabbing anything within range that moves. For this dragonfly nymph, the strike is slowed down fourfold as it feeds on a damselfly nymph. But a high-speed camera is needed to show the details of the strike. Slowed down even further, you can see the hooks on the side of the labium open wide, then snap shut to trap the prey, which is then drawn back to the chewing mouth parts. Nymphs may lay in wait, as in these examples, or may actively stalk down their prey. Early-stage nymphs are semi-transparent, allowing you to see into their bodies. Look how this unfortunate prey, still writhing, slides into the nymph's gut. And that set of bumps in the center of the nymph's head between its eyes, that's its brain. After many months and many molts, the adult, still trapped in its aquatic skeleton, climbs out of the water to undergo the final molt. Most nymphs climb up the nearest blade or stem, emerge from the exoskeleton, leaving behind the empty skin, and climb further to dry and harden. And eventually, while still pale and weak, they fly away. Some species crawl onto a convenient floating platform to emerge. The adult first breaks its head and back out of the old skeleton, arches its back to loosen the abdomen, then finally pulls the rest of its body out. It then pumps its blood into its wings and abdomen to expand them. Sometimes the pressure forces a drop of liquid out. Finally, its wings partially harden, the full adult form is reached. At this stage, dragonflies are soft and vulnerable, a delicacy, to birds at least. So as soon as they can, they fly off to hide away from the water and mature into the acrobatic, colorful adults we see all summer long, allowing the species to reproduce and disperse to new bodies of water. So, head out and enjoy them. Hopefully you'll find them even more fascinating now that you recognize their unique behaviors and understand their complex life cycle. They're creatures you can enjoy for a lifetime. And when you're out, enjoy the other surprises you're likely to find, like this song sparrow bathing, or minnows lurking under the surface of a small stream with water striders gliding on top, or these skipper butterflies feeding on the nectar of an arrowhead plant. Even plants can be amazing, such as this fully submerged aquatic plant that once a year pushes flowers out of the water to be pollinated by insects. And what have we here? Ah, more dragonflies.
I hope you enjoyed that video. I really enjoyed making it, but that wasn't my purpose for being here at Fermilab. My purpose was to monitor the dragonflies and damselflies here to find out which were present, where they were, and how abundant they were. I was trying to help evaluate the LMUs that you saw earlier by locating the highly productive areas and contribute to the assessment of the relative sensitivity of these areas in conjunction with all of the other species that are being studied here. I conducted the survey from mid-2012 to mid-2017, 5 seasons equivalent, and visited a total of 38 sites and found 58 species of dragonflies and damselflies, about half of each, 30 dragonflies, 28 damselflies. The data was entered into a single spreadsheet that would facilitate analysis of the data later on, and also was compatible with a large access database that lists all of the species on site. This is the sensitive areas map that was constructed in 2015. It's being updated right now. And you can see that most of the habitat is colored green. It's a conservative approach. Ryan Campbell, the former ecologist at the lab, said, not everything can be dark red. The sensitive areas are highlighted by 3 colors, yellow, orange, and dark red, low, medium, and high sensitivity, which guide the input that the ecologists make to the people at the lab as far as development of the lab, and also guides the maintenance of that lab, of those areas, by FNA and the lab crews. There are 3 approaches to monitoring. One is transects, which is like a time-lapse video. It's where people walk through an area on a defined route, observing everything within a 20-foot bubble and record it methodically several times a year. It's a useful approach for finding common species and looking for changes over long time periods where you can see effects of urbanization or climate change. But another approach I call saturation, where you're trying to see everything you can. You would go out with binoculars, with telephoto lens, stop. It's like having a bunch of trail cams with you that you set up and then scour the pictures at the end of the season. And a third concern is breeding. Are the species actually completing their life cycle on site? So analogous to breeding birds, where people would look at courtship, nesting, and fledging, with the dragonflies, since we know there's no courtship, except in a couple cases, it would be mating, egg laying, and emergence. Here's 2 examples of surveys I did. The first on the left is a very preliminary survey by bicycle, where I tried to search out the various different areas of the lab, going in the public areas and bicycling through the private areas as well. I'm showing this in part just to remind you of how big the site is. On the right, you see a survey by foot, which is typically what I did. Almost all were on foot. This one's a little longer than most. It goes past 4 different areas: Kingnut Woods, Prairie 24. Then in the center, the squiggly area, that's the wetland mitigation area. It's one of the most productive areas in the lab, not just for dragonflies, but for other things as well. It carries into the stream, Indian Creek stream, going into Indian Creek Woods, which is a much less productive area. Like I mentioned, all this data was, oops. Sorry. All this data is entered into a single spreadsheet where the important entries are the column shown in red. I made it codes, simple little codes that I could remember for the location, the LMU, and the species, and then would record the count and any reproductive behavior observed. Those codes are translated by a linked spreadsheet to generate the detailed information, where the LMUs are spelled out and the species are given their scientific and common names and their taxonomic classification. This page also includes two indicators of rarity or commonness. One is status. That's a ranking system out of the Science Museum at Springfield for the state of Illinois. S5 is a very abundant, well-distributed species, totally safe. And S1 would be a state endangered species. And on the right, the last column, percent DMN sites. That refers to the Dragonfly Monitoring Network, which was conducted over many years previously to my studies, and which I participated in for a while. 3 years of that study, from 2008 to 10, had particularly large data sets with over 60 sites on average being surveyed and a total of 80,000 plus observations. What I did was took that information, which was available on spreadsheets, and calculated at what percentage of the sites was a species seen for every species I saw. So across the top, you can see the eastern forktail. It's the most common species on site, S5 status, and reported at 76% of the DMN sites. But down lower, there's another, the comet darner, rated S2, and observed at only 3% of the sites. This is additional information that is brought into consideration of the raw numbers that the results generated. But what a table like this does is it lets you generate many complex or simple outputs from all the data from the 5 years. And here's an example of a complex output. This is a classification or summary of all the dragonflies observed and damselflies halfway through the survey at the time of the making of the LMU sensitive areas that you saw earlier. And you can see which family, this is all in taxonomic nomenclature, Anisoptera, Eschneidy, the scientific names. But you can see that 4 dragonfly species families were found and 3 damselfly species. See which the individuals are and the number of individuals seen. It's a very rich set of data. But a simpler set that gives a more concise bit of information is simply to make a graph, a chart of the number of individual species tallied per site. The number of individuals of each species observed. And the tallies ranged up to from 1 to 781 for the eastern forktail. But the important thing to note is that just over a third of them dominated the abundance, the populations that we're seeing here. And in fact, for 25% of them, out on the right, there were 10 or less seen, and for several, only 1. A more relevant chart generated from a different table is the number of species seen per each area. And here you see the numbers range from 38 down to about 2. I ranked these fairly liberal as a starting point, saying that areas that had greater than 15 species would be considered productive areas. The graph itself, the table itself that generated that graph is shown here with those 15% or 15 species or more listed, but also a few extra. One thing that's important to note is that Mason Hill Ditch, a non-LMU, was a highly productive area. That's the ditch that I showed you earlier in the presentation. It's also part of the cooling water system entirely. It has nothing to do with the natural water that was on site originally. But there's some considerations that have to be brought into this. Is that one thing is that with so many sites and so much space, it wasn't possible to monitor each site an equal amount of time. In most cases, they were close enough that there shouldn't be any significant differences in this ranking, but there are 4 on this list where I think there are. The first are the second and third, the Ed Center Pond and what's called Kidney Pond. That's the pond right next to the Wilson Building, straight west where the parking lot is. Those were oversampled. They were easy to get to, but I deliberately oversampled them because they're part of the public areas of the lab. The Letterman Science Center is next to the Ed Center Pond, and students come there frequently and sometimes go out and actually survey the aquatic species on the pond. So I wanted to have better information for these areas that could be part of educational programs. But if all those extra samplings were dropped, they would only drop a few positions, because when you sample, you get a curve like this. It's sort of a hyperbolic curve. And if you climb up here, you sample 40% more time, you only climb a little bit on that curve. The other 2, though, that are undersampled are near the bottom, Nepesi Marsh and Site 29 Woods. Those are both fairly interesting areas, but both have residents. At Nepesi Marsh, there's an osprey nest that nests there most years, and the director lives in Site 29 Woods. Both of them deserve a little privacy, I think. Another consideration that should be brought in is whether the species reproduce successfully on site. And here are the three levels of criteria: the mating, the egg laying, and emergence. With emergence, there are two different bits of evidence: the shell the shells that the larvae produce when they break out and emerge, which are called exuviae, and the pale, weak, flying, uncolored youngsters, which are called tenerals, that have just emerged. For 42 of the 58 species, at least one of these was found, which is around 70%, and for a few, all three. Another consideration, or an application of that consideration, is looking at two species in particular, the the comet darner and the flag-tailed spiny leg. Both of them were seen at only 3% of the DMN sites. Both were somewhat down in the ranking by the state, but the reproductive behavior, the habitat, and the sighting itself were dramatically different for those two. On the left, you see the comet darner. This is a photograph taken elsewhere, flying across a pond much earlier, but it's exactly what I saw at the Letterman Science Center pond. You can see where the comet darner gets its name from that bright red tail, but it shot past and didn't display a hint of interest in the pond below it. And I'm pretty sure it just continued going off-site to some other place. But on the right, you see a flag-tailed spiny leg teneral. Remember the photograph I showed you in the beginning of a female spiny leg by the edge of a pond? A year later, on the opposite side of that pond, this individual flew up weakly and landed in the shrubs and let me get close enough to take shots. If you look here at the end of the abdomen, you can see the large club shape turning down that looks like a flag that gives it part of its name. Up here, you can't see it without enlarging, but I could enlarge, and you can see the spiny legs. So this is a definite completion of the life cycle on site and should be weighed into the consideration of that pond. Whereas I would feel the comet darner should just be struck off the list. It has nothing to do with dragonflies living at the lab. A final consideration that I need to add to the talk, and people add for evaluation of other species, is how suitable is the situation in which you find species. These are two species you've seen earlier, the citron forktail on the right and the western red damsel on the left. Both of them prefer the same type of habitat. Both were seen in that habitat. They're both rare in the Chicago area. And that area should be treated a bit differently because of the presence of these two species. But fortunately, in this case, it's already treated quite well. It's the wetland mitigation area, one of the most highly ranked areas on site for all characteristics. So here's the map again, and all of the groups involved are now putting in their data to tweak the information here. I don't expect that there will be significant changes. There may be one or two that move one level in the hierarchy, the different tiers, and maybe some areas added as sensitive areas. But a lot of the information that's going to be presented is going to be very useful for guiding the maintenance and improvement of these areas. And the input comes from all sorts of things, primarily plants, followed by birds, and after that there's bees, butterflies, damselflies, salamanders, frogs, snakes, turtles, even bats, and flying squirrels. It's quite an impressive effort, and one that I'm glad to have been involved in. So I'd like to end by just making a comment about Fermilab and its relationship between the natural areas at Slet-Furch and its goal as a particle research lab. In all cases, the experiments come first. This is a screenshot from a Fermilab promotional video or informational video about DUNE, the Deep Underground Neutrino Experiment. It's a major international experiment under construction at Fermilab. And at the bottom right, you can see a building being built. That's being under construction right now, where the particles are initiated. They're accelerated out, circle around, and at the top center, leave as neutrinos. These neutrinos go underground for 800 miles to a detector in western South Dakota, and they have to go straight, and they have to leave from there. So there's no choice. And if a ecological functional unit, a feature, gets in the way, it has to go. But back here, the other aspect of Fermilab's treatment of the environment is that this pond here isn't just an artist's concept. It's actually a cooling water pond that will be part of the system. And the plans for the very end of the construction is that that pond, the edge of that pond, will be planted with native wetland species. There's going to be no problem finding people to maintain that. There'll be a cadre of FNA volunteers to help. And I plan to return to dragonfly monitoring there to watch the pond get colonized and emphasize more the nymphs and their emergence than I did before, while also monitoring the adults. To learn more about ecology at Fermilab, you can visit the website ecology.fnal.gov. It's a very rich site with deep links, and down here, wildlife conservation. If you plow in there, you can find my page on dragonflies and damselflies. And next to that, talking about volunteerism, is a photograph of one of the FNA workdays at the end of the day. To learn more about FNA, you can visit our website, our Facebook page, or email us. This is a very striking screenshot from our Facebook page with a prairie burn. I should state that we volunteers don't do the burns. That's left to the pros, but we can sure photograph them. They're quite impressive and critical to the maintenance of prairies. The lab's closed now. You've heard that. I'm sure you know that. But if you want to find out as soon as it opens, sign up for our newsletter, because when that happens, that newsletter is going to come out telling everybody. And I'd like to end by thanking everybody who made this possible. First, as far as dragonflies go, Tom Vallot of the Forest Preserve District of DuPage County got me started monitoring dragonflies. And Craig Stettner managed the DMN during the period when I monitored for Tom. He single-handedly collated the data of all of the volunteers for those 60 plus areas I told you about and created a useful Excel file from it. He also introduced me to Marla Garrison, who mentored me for several years, primarily on on damselflies. She's created a great field guide to damselflies, and then on nymphs. At Fermilab, Jacques Hoymans, an old friend, we ran into each other and he suggested I come into the lab and get in touch with Brian Campbell. Jacques also instructed me how to do the Excel spreadsheet, and with the help of my wife, Vita Bonnewell, a former colleague of Jacques, they forced me to do it, and I'm very grateful to them for that. Ryan let me into the lab, welcomed me here openly, and encouraged me to seek out every area I could find, and actually mentioned several others I didn't know about from the literature and maps that were available. Wally Laverniere, the current Fermilab ecologist, has been equally supportive of my more recent activities, making educational videos about stewardship at the lab, primarily for the Letterman Science Center, one for them, and a couple for FNA. And finally, FNA itself, for their creating the infrastructure that let me monitor, and more recently for tolerating my intrusion into their workdays just to video them. I'd show up and they'd suffer me. So thank you to them. And with that, I'd like to thank you for listening to this video. I hope you enjoyed it, and turn it back to Sue and answer any questions you might have.
Well, thank you, Mark. So I invite people to write some questions in the chat box. You got very many comments on how beautiful your photography was, Mark. So too numerous to mention. But while we're waiting for some more questions to come in, here's a question. So how far do some dragonflies and damselflies migrate?
There are some species that migrate quite long distances. The damselflies aren't a strong flyer, so they migrate less. They may travel on wind. But the green darner, the species that we see so commonly, has been known to be migratory for many years. Studies on the East Coast have actually put telemeters on them and followed them with Cessna airplanes as they migrate down the New Jersey coast. The black saddlebags is another migratory dragonfly, and there's an organization, the Xerces Society, that studies insects and includes a section that specifically addresses migratory dragonflies and damselflies. So you may want to look at Xerces Society. forget the name of the actual project, whether it's migratory odonates or something like that, but yes, several do migrate. Some of the species that we see here may just be passing through. Whether they are at the edge, if they're at the edge of their range, they may not successfully reproduce here, but yes, they migrate.
Okay, thanks. And here's another question. So what can you tell us about the feeding habits of those odonates that mature in seeps?
I don't actually know what this person may be asking about the larvae, I suspect, because the adults feed on other flying insects. I would imagine that they're feeding on any small crustaceans, anything in the water, worms. They will. I've tried to raise some, and if you put anything that moves in there, they will snap it up and eat it.
Okay. And then a question about how long do dragonflies and damselflies typically live?
As I mentioned, the adults live for only a few weeks, and that can vary, maybe say 2 to 4 weeks. But the larvae, the nymphs, can live for quite a long periods of time. The one famous one is the Heinz emerald, the federally endangered dragonfly. It spends 5 years as a larva, and its larval home is the crayfish burrows. So that's probably why it's endangered. It needs the habitat that reliably supports crayfish, and then it needs a long period of time to make it through that development cycle. In northern climates, I've read that's way up north, where it's cold, some species will spend 7 years as nymphs in the water.
Okay, and here's a clarification on the question about seeps. So it is about the larvae, and the question is, are the larvae visual hunters?
That I don't know. I don't know if they are tactile or visual. They could be either in that situation.
All right. And then a question about the eggs. We saw that they were laying an awful lot of eggs. So about how many of those eggs end up being adults?
That I don't actually know that at all. That's the success. I once reared some, and rearing is harder, but I once hatched 50 eggs of a species, and one made it to the last larval stage. But in nature, it could be like that or worse. Many of them are going to be eaten. They are prey as well. And the cycles that they go through in maturing, I think there must be a great attrition until they've actually established themselves. I have surveyed the damselfly populations at the Letterman Science Center pond in the fall and the spring to see how much they change. And during that period, they're quite stable. But I don't have a number on any estimate of how many eggs were there initially.
Okay. So are there any more questions? We have time for the recording of the full talk will be available so that you can see that again. And there is a question here of, do you give presentations at groups?
I haven't, but I have small groups. I've done presentations for FNA, and I've retired, and if it's close by, I'd do it. If it's a long ways off, I might not. I'm real selective now, but yet I would be willing to do presentations for people.
Okay, that's great. And then another question. Do either adults or larvae of any species have specialized diets?
If the diets that I could imagine are specialized would be specialized because of their location and their availability of food. I think not. I think they eat anything that they can get their mouth parts on.
Okay. And then a question about whether the populations are stable or are they declining? And if declining, why?
most insect populations have declined. And I recently read an article in Science that surprisingly said that looking globally, the aquatic insect populations weren't suffering as much as the terrestrial insects. In my experience, I've seen booms and busts here, but in general, insect populations are vulnerable, they are declining. And I was surprised to read that, the description saying that aquatic insects weren't. I don't recall the breakdown in that article, whether it was broken down into the different orders of aquatic insects, but they insects in general, yes. Aquatic insects, less so.
All right, thank you. Well, we're about at the end of this presentation. I don't see any more questions, so I want to thank everyone for attending and invite you to check the Ed Center website for Children's Earth Day activities, which will happen on Thursday, and I believe we're going to get that link into the chat box if you're interested. So once again, thank you everyone, and have a good rest of the day. And thank you, Mark.
You're welcome. I enjoyed it.